cancer immunology and immunotherapy is really, really exciting because it's showing some really remarkable responses. And sometimes for immune checkpoint inhibitor, patient can be completely cured. Say, already metastatic tumor, patient has failed many different drugs already, suddenly on immunotherapy, and after some 10, 20 dose injections, or then they're completely cured, 5, 10 years, nothing. It's like tumor clear. And because of this, the scientific community, pharmaceutical industry, biotech industry, all really pounding on this. And all the new technologies are thrown to cancer immunology. And I'm hoping that you can see how computational biology can really play an important role in here. Okay, so I will show you what are the problems in the cancer immunotherapy or to study cancer immunology. This is the original figure we show, T cells with the T cell receptor and the cancer cells presenting the antigens to be examined and the PD-1, PD-L1 interactions, things like that. So we look at each step. The first step is what mutations are in the tumor. Obviously, if the cancer cell has mutations that are easy to recognize, they will actually respond better to immunotherapy. And this is more or less related to Sam's question. For some pediatric tumor that has very well-known one mutation, do they work? It could be that they don't work so well because for some of these pediatric tumors, the patient's immune system didn't develop correctly in the first place. And that one mutation may not really have an immune response. Whereas when you have a later stage, like adult, which has initially an okay immune system, then immunotherapy actually can work well. And so this one has been found to be a pretty robust biomarker, which is tumor mutation burden, or TMB. In general, remember with targeted therapy, patients who have more mutations, they are hard to treat because if you have a targeted drug that really target one mutation, it can easily mutate to block the drug from binding or evolve to have other mutations which evade this first gene that you're targeting. They can start to grow based on some other driver genes. And so traditionally, the higher mutation, the harder for this cancer to be cured. But in immunotherapy, it's kind of opposite. The patients with more mutations are more likely to see a response benefit to at least PD-1. I think for CTLA-4, this is the same. Because you can imagine if your tumor has like 100 mutations instead of 1, there are more chances for different T cells. Say if that guy with the right T cell to recognize mutation A is dead, this patient still have 99 other mutations which can potentially be recognized by the T cells. They are more likely to be recognized by the patient's T cells. And so in general, this is one case. You can see here, the y-axis is the exon mutation burden. And each is a patient, right? And for this, you can look at their response. So PR means partial response. CR means complete response. Complete response means tumor is completely gone, which is great. Partial response means the tumor has shrank by 30% or more. Stable disease means the tumor is not really shrinking the tumor, but at least the tumor is no longer growing. Whereas PD means progressive disease. Tumor is still growing, right? So you can see here, when the patient has more mutations, they have more partial response or stable disease, whereas when they have very few mutations, they have more progressive disease. Okay, so that's one. You can imagine how we do this. You take the tumor out, and also get the blood, you do whole exome sequencing, you look at their mutations. And out of those mutations, which mutation are likely immunogenic? It's part of your homework 6. So there are some mutations which change amino acid sequence, but there are other sequences which only change on the DNA, but they don't change the amino acid sequence. Which of those are more likely to be immunogenic? they need to, yeah, if they change the amino acid sequence, they are more likely to be immunogenic, right? And so you can imagine, computational algorithm for mutation analysis. First, you call the mutations, you look at which one actually change the amino acid. Those are definitely important. However, just because this mutation is changing the amino acid, it does not mean it will be presented onto the cell surface. And this is because of this antigen presentation issue. So the gene responsible for this is called the human leukocyte antigen. The protein complex is called the major histone compatibility. And there are two types on our cell surface. Do I have a slide? I should have, yeah, maybe I should have done this here. So as we mentioned, within our body, we have two types of antigen presentation complex. One is called MHC1. All of our cells have MHC1, which routinely present whatever its own degraded protein onto the cell surface. MHC2, as I mentioned, so this is MHC1, and if it's presented, our CD8 T cells will recognize it. Whereas MHC2 are usually on professional antigen presenting cells like B cells, macrophage, or dendritic cells. Macrophage is really, it can just eat things. That's what macrophage does. If it sees some bacteria, it can just eat it up. And so ingest this in here, chew it up, and then present it onto the cell surface. It's to help other immune cells to recognize, usually memory cells. So these are CD4 T cells. They're also important. And so why are these important? If you look at our body, the highest difference, if you do SNP typing, every two individuals, they are similar by 99.999. The very small part that's different, a lot of them are different by this HLA gene. On this, it's different, depending on your race, your ethnic, there would be differences. Even the same family members could be different. And so in fact, if you want to do a bone marrow transplant or some organ transplant, you need to make sure their types match. So for example, if there is one patient, you have two copies. So if there's HLA-A, HLA-B, HLA-DR. So these are the three most important HLA loci. And you have one copy from mom, one copy from dad, and then the donor has also one copy from mom, one copy from dad. You want to make sure those 6 loci all match. If they all match, you can have a organ transplant or a bone marrow transplant. Whereas, if you have 5 out of 6 match, it's not ideal because your body can reject the organ. Or if you have a bone marrow transplant, you could have something called graft versus host. The bone marrow you got is rejecting your own body. That's also bad. Okay, so you make sure that they are really matched. And so because every of our cell has also, by the way, MHC1 is presented by all of our normal cells, whereas MHC2 is only, it's mostly highly expressed on the dendritic cell, CD4 T cells, B cells, and macrophages. Because these are kind of expressed, you can call them from your DNA sequence or RNA sequence. You can do that from sequencing. And previously, people have already matched this. So for example, this is one algorithm called polysolver, another algorithm called OptiType. They both can do this pretty good job, both on the DNA and on RNA. Polysolver has additional function which can detect mutations on the HLA. So for example, so if you have the whole exome sequencing, you can use some whatever alignment to align them back to the genome. And for once you have this, because we know what are the different HLA types, and you based on the read alignment, you can make your best guess. This patient has what's the one allele, what's the other because you have mother copy and father copy. You can genotype to make to know what their the 6 different. So HLA-A, B, and a DRB1. What's the two alleles there? Interestingly, they can also look at the reads that's mapped to the HLA. So the red reads, I guess, is related to the HLA. The blue reads are mapped to some other regions in the genome. But you can see in this case, say from the germline, from the blood, you know the HLA type, you know the A, B, and C, the two copies. But then from the tumor, sometimes you can see when you do the alignment, you notice, oh, there is a mutation on this patient at this HLA locus, right? Can you, yes, Jet?
What's the reference?
Because you have the blood.
Oh, you use blood.
Yeah, so you compare the tumor and you can see, there are some bases that's different between the blood, the germline blood, and this is from the tumor. And there's a base that's mutated. So why is that mutation important? So I'll show you, for example, so all of our normal cells have MHC. Different MHC bind to different peptides. So every set of HLA, like they bind to a different peptides. And that binding looks like a kind of a protein motif. Remember transcription factor motif? So HLA is like a protein. It recognizes other peptides. And so you can write this motif out as, do you remember what this is called? It's called a sequence logo. So this is one HLA type that can recognize one sequence logo. I'll show you. So the different HLA type, say you can compare like Caucasian or like African, or you can compare Asians, they will have different HLA types. And so they will recognize different peptides. So when our proteins are degraded, usually there are enzymes that cut at like lysine or some specific location to make them into like small peptides. But depending on what is on that peptide, some of them will be presented by the Asian HLA, but not presented by the Caucasian HLA. So even though the same protein get degraded to chop up into small fragments, depending on your HLA type, different things will get presented onto the cell surface because you can see they recognize very, very different peptides. They favor different things. So can you guess what happens when you have mutations on the HLA gene? Why is it mutation? So for example, if you have a mutation, it normally is presented onto the cell surface. One way for it no longer to be presented is if you have a mutation on this particular HLA, then that mutation is invisible on the cell now, right? Then your T cells can't see it. And so mutation on the HLA is a way for the cancer cell to evade your immune detection. Okay, that's a very useful thing to kind of learn. Yeah, so also sometimes cancer's mutations can also, so you can imagine, recently there are papers to show that if you look at all the mutations on the cancer in general, they tend to appear on regions which is not presented by the HLA of that type, of that patient. Because if it's presented, it will very likely be already recognized by the immune system and that cancer cell will be wiped out. So in general, whatever is left in that mutation, in that tumor, at least in the very early ones, that really gets, so you can imagine when somebody just had a mutation in very early on, if it's immunogenic, your immune system will wipe it out. But so instead, what stayed were those mutations that don't normally get presented by this patient itself because the HLA type they have. And so you can see, depending on whether you're Caucasian or whether you're Asian or African, your driver mutations could be different because very often the driver mutation is not presented by their HLA type, something like that. Does that make sense? Yeah, so they have different preferences. So you can see, depending on the individuals, there are 2,500 different HLA alleles, although there are some really common ones. I think the Han Chinese or the major Caucasian, they have some very, very common HLAs, but there are like 2,500 different ones. Currently, we only know, but like 50 or 70, those a limited number. The most common ones, what is their binding preference, very well. But for the rare, small village in some ethnic groups, we may not know their HLA type very well. And so those we don't do a very good job predicting. And also, there are also, our current algorithm only predict HLA, MHC class 1, so self-presenting, those are predicted well. Cross-presentation from the antigen-presenting cells, which, two things happen, spit it out, those currently there's no algorithm that predicts this very well. Okay, so there are computational algorithm, it's called NetMHC, and depending on HLA type, they tell you, what things can get presented. And the stronger the binding, the stronger the antigen presentation. Okay? So that's the second part. Maybe we'll have